this is where we're supposed to wait before we go on, right?
Yeah, I think this is the right place.
Okay, cool. Hi, I'm Sydney Florianzia.
Danny Powell, nice to meet you.
Likewise. I'm working on my PhD in chemical engineering at the University of Washington.
Oh, you're in chemi? My dad's a chemical engineer.
Oh, no way. Are you also a chemical engineer?
No way. I'm in molecular biosciences. My brain cannot do that engineering stuff. I'm actually at Washington State University in the Immunology and Infectious Disease program. So how did you get into chemical engineering?
Well, for me, it actually started with music. So I was at an engineering camp in middle school where they taught us all about how music and neuroscience and engineering were actually all linked. And I just became so intrigued by the intersection of art and STEM.
Oh, really? I'm into art too. You know, for a long time as a kid, I thought I'd either be an artist or a scientist. And I mean, I still like drawing, but science won out in the end. In high school, I saw this really cool documentary about CRISPR-Cas9, an exciting new gene editing technology. And it was just so interesting, I immediately knew I had to be in bioscience. And I was hooked. But chemical engineering and neuroscience, what do you do with that?
So I study the blood-brain barrier. It's the collection of cells and proteins that line the inside of the small vessels in your brain all over. And so I look at how each of the cells that make up the blood-brain barrier contribute to letting medicine into the brain or keeping medicine out, and how all that changes in disease. So really looking at the transport of different medicines from the blood and into where it has to go in the brain. What about you? What are some of the things that you study?
So I study the tick-borne diseases Lyme disease and relapsing fever, which are caused by this little guy, Borrelia. I'm actually really interested in how they regulate their ability to evade the immune system and cause these debilitating diseases. I do this by looking at a particular segment of the genome that is responsible for their ability to evade the immune system. We identify key regions of interest and remove or change that DNA to develop a pool of mutant bacteria. And then we test that bacteria for its ability to cause disease in a mouse model.
Okay, so I know a bit about Lyme disease, but what is relapsing fever exactly?
Well, it's endemic to the inland Northwest, and it's characterized by a nasty fever that lasts a couple days and then repeats or relapses every week. We're hoping to identify potential antibiotic targets that are specific to Lyme disease and relapsing fever, so we can move away from our dependency on broad-spectrum antibiotics like doxycycline and penicillin, which not only wreak havoc on the body's natural microflora, but can also introduce antibiotic resistance into the population.
Whoa, that already sounds impactful, but what are some of the other impacts of your research?
Last I checked, the CDC reports 30 to 40,000 new cases of Lyme disease each year. And then if you take into account all of the misdiagnosed and unreported cases, the real number may be 10 times as high.
Oh, wow. So it sounds like there's a lot of people that could really benefit from better treatments for Lyme disease.
Oh, for sure. I think the annual healthcare cost in the United States is in the hundreds of millions of dollars, possibly $1 billion a year. So you're looking at medicine for the brain. What kind of diseases are you trying to treat?
So for a lot of brain diseases, the challenge really lies in getting the medicine where it has to go. And so while the things that we study in our lab can benefit a wide variety of diseases, we specifically study the diseases caused by neonatal hypoxia ischemia. So basically when a baby's brain doesn't receive as much oxygen close to the time of birth. And so unfortunately, this contributes to 8% of childhood deaths worldwide, with that number increasing 2 to 3 times higher in low and middle income countries. So while it's a challenging problem and an interesting problem, it's nice to know that the work that I do has the potential to positively impact a lot of people around the world.
That's really cool. Are there a lot of labs that do this kind of research?
Very few. So when I was an undergrad, I was actually recruited into a neuroscience lab because I was a chemical engineer. They needed somebody to build and load their microfluidics, which are basically these very, very tiny channels that we load living cells in to build a blood vessel in the brain outside of the body to be able to test and see how drug delivery might change in different diseases. And so with my experience there, I was actually recruited to my current lab at the University of Washington, the NANCE lab, which allows me to do the neuroscience that I want to do from a chemical engineering perspective. And then when I got the ARCS fellowship, it was a no-brainer to come to the UW. And so I really knew somewhat about the science that was going on, but I didn't know that I would be entering such a vibrant scientific community, both in ARCS and Seattle overall. How has the ARCS fellowship benefited you?
Well, I'll second that thing about a vibrant community for sure. I've had the wonderful opportunity to network with fellow researchers at WSU and at UW. And the financial benefits are, of course, very nice. I can be stressed in the lab, and then I come home and I do not have to be stressed when I pay the bills.
That's definitely valid. The ARCS fellowship allowed me to create a place that feels like home in Seattle. I'm actually really far away from my home. My hometown's in North Carolina. So the ARCS fellowship really allowed me to build a space in my apartment that feels like home. What about you? Are you close to home?
Yeah, I'm pretty lucky in Pullman to only be a 2.5-hour drive away from where I grew up in the Tri-Cities. I actually graduated undergrad at WSU as well, and then I stuck around and joined a lab on campus where I enjoyed the research so much that I stayed as a grad student.
Okay, so we've talked a lot about our research because it's kind of our whole lives. But what do you do outside of the lab?
Well, to keep myself sane, I provide myself with enrichment in the form of movies. I actually go to the next town over, Moscow, Idaho, where there's this wonderful historic one-room theater that does curated film showings. So just this year, I've gotten to see movies like Apocalypse Now and Pink Floyd's The Wall in the theater. It's really special. You're over on the west side, right? You must have a lot of opportunities for enrichment in Seattle.
Yeah, being so close to Capitol Hill, it really is a choose-your-own-adventure. There's always something happening, something going on. But one of my favorite things to do is actually at UW. I take myself on what I call flower walks, where I go and just look at the nature around campus. And it's really cool because I can take the same walk 1 to 2 weeks apart, and there are different flowers that have bloomed. So when I have a long incubation in lab, it's a great thing to go do. I also do a fair amount of STEM outreach. So just like I was ignited to do science at that engineering camp back in middle school, I try to light that same spark in STEM for other kids and young folks.
That's great. That sounds like a wonderful way to give back to the community.
We're ready for you now.
All right, you nervous?
A little, but I'm excited to talk to everyone.
All right, well, let's get out there.
Alrighty.